Welcome, cousins near and far. Today we're going to be discussing chromosome mapping, or as some of you may prefer, chromosome painting. And we're going to answer some of your questions. What is chromosome mapping? How do you get started? And what is the purpose of it? To get the most out of this video, you're going to want to be familiar with the basic terminology of genetic genealogy. I have a short series, Getting Started with Genetic Genealogy, that I would highly recommend you viewing. I'll link that series in the description box below. That said, let's chat about chromosome mapping. What is chromosome mapping? To answer this effectively, it's important to first know a few of the basics of genetic genealogy. Firstly, humans have 23 pairs of chromosomes, and I'll come back to that in a minute. So, humans have 23 pairs of chromosomes, and when dealing with genetic genealogy, you can almost think of it like computer coding. We are essentially looking at 23 lines of coding. This coding is made up of genetic data that you inherit from your ancestors. Lines 1 through 22 are your autosomal DNA, abbreviated as ATDNA. This is randomly inherited DNA data, roughly 50% from your mother and 50% from your father, given to them from their parents in the same fashion, as from their parents, and so forth. This DNA data is little snippets from thousands of your ancestors back to the dawning of our species, continuously split up and randomly inherited each generation. To understand this better, take your mother for instance. Your mother was 100% whole, a whole person. You are only inheriting 50% of your mother's DNA. Understanding basic math also tells you that that's 50% of your mother's DNA that you did not inherit. You keep hearing me say random inheritance, and this is important for a few reasons. Firstly, it's wildly beneficial to test siblings, aunts and uncles, cousins, grandparents, anyone you possibly can in that close relationship range. Take a full sibling, for example. Since you only inherited 50% of your mother's DNA, it's not just probable, but expected that your sibling inherited a different 50% of DNA from her. The data inside the different DNA segments inherited is why you look different from your family members, just as is the reason you look similar. Another reason random inheritance is important is understanding that we have branches of ancestors to which we will not inherit DNA due to the randomness of the DNA we inherited. Consider your grandparents. You have 4 biological grandparents, 2 from each parent. In a true mathematical cut, if you have only inherited 50% of DNA from each of your 2 parents, then that means you've inherited only 25% of DNA from each of your 4 grandparents. 25%. That's 75% of DNA from each grandparent that you did not inherit. Another point to consider is that when we inherit our 50% of DNA from one parent, that randomness is not evenly split from what in turn came from each contributing grandparent. It's random. So you very likely could be inheriting more from one grandparent over the other. Now, think of all those ancestors whose DNA you did not inherit. It doesn't make these ancestors any less your ancestors. You still came from them biologically. You just didn't inherit their genetic DNA data. Now think of all those people wondering where their Native American DNA is, their Mediterranean DNA, their Germanic or Scandinavian genetic inheritance. It's in their family trees, so why isn't it showing up in their DNA? This is the reason. Now, so far, we've discussed autosomal DNA, which you inherit on chromosomes 1 through 22. This DNA you can have tested at all the leading DNA testing companies: Ancestry DNA, Family Tree DNA, 23andMe, MyHeritage, Living DNA, none of which I am affiliated with. These autosomal tests can be taken by both males and females and are fantastic for bolstering family tree research and finding relatives for all your branches. Of course, getting DNA matches is heavily contingent on having relatives that have also tested. There's a lot more I can say on the topic of DNA testing and the company specifically, but that's perhaps for another video. So let's discuss that 23rd chromosome pairing. Chromosome 23 is your sex chromosome. It's responsible for you being male or female. The letter Y represents the male DNA, and letter X represents the female DNA, also called mitochondrial DNA. A male possesses one Y from his father, which makes him male, and one X from his mother. A male can pass along to his child either his Y or his X, determining if the child will be male or female. A female possesses two X's, one from her father and one from her mother. A female can only pass along an X, as she only has two X's to give. So, at birth, you are assigned one sex chromosome from each of your parents. If you are male, you have a Y and an X, and if you are female, you have two X's. This Y chromosome and or X chromosome, which you've inherited, is the direct paternal and or maternal line, unbroken, going back in time to the dawning of our species. If you are a male, you have the Y DNA from your father's 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 father's, etc. line, unbroken, back through time, and the X DNA from your mother's mother's mother's, etc. line, unbroken, back through time. If you are a female, while you have the X from your father's mother's unbroken line, you will always present with the mitochondrial haplogroup from your mother's 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 mother's, etc. line, unbroken, back through time. And we'll discuss the whys of that a little bit further in the video. So, how does this relate to the region or ethnic breakdown? Unlike autosomal chromosomes that change rapidly each generation due to random inheritance at each generational level, the Y and X chromosomes change or mutate at a much slower pace. So instead of having mutations or changes in the genetic data each generation, as we discussed with the random inheritance of the autosomal DNA, these changes happen very slightly every few generations. This is where haplogroups come into play. Much like our family trees, imagine a great-grandfather that has multiple sons, each son migrating to a new location and having their own children. One group of descendants might go to the mountains in the cold north. Another group might go to a warm island in the south. Another might be farmers in the plains. Each group comes from the same origin point, the same forefather. But as they migrate, they must adapt to their locations, and so make changes to their lifestyles. Think of a recipe that this family may have had. As these sons went to their new locations, family recipe in hand, they ran into a problem. Not all of the traditional ingredients were available, so they had to substitute certain things, thus changing the family recipes a small fraction. When we look back in time, while comparing these different versions of our family recipe, we can see which ancestors changed that recipe. We know his recipe started out in its original form at a specific location, where it can still be found today. Researching our history and the events in the region, we can get an idea of the whys and whens our ancestors may have migrated. And as we see that ancestor settle into his new region, we understand why he changed the ingredients to his family recipe. We can compare all these new recipes for these family branches and track them back to the original recipe, the common ancestor. We know the regions where the changes occurred in the recipe and can begin to see the migratory patterns of the family through time. While we have no control over the mutations that occur, and they are not influenced by our geographic location or culture, we can ascertain via DNA testing pools or testing the oldest members of the regional community and coupling that DNA data with known migratory patterns of the area. When our DNA is compared to these data pools, we are given a percentage of likeness of DNA data in comparison to the DNA of the data pools, and so are presented with a tentative percentage of how much DNA likeness we have to those oldest members of a regional community. We're doing the same thing on chromosome 23, the sex chromosome. Because these changes happen at such a slow pace, and the chromosome is passed along virtually unchanged for generations, we can actually discern which mutations occurred in a specific generation. This is such a predominant feature that when you have your mitochondrial DNA, aka X chromosome, or the Y DNA test done, it makes more sense for the labs to compare you with the original beta source or a base recipe and present you with a listing of genetic mutation markers. These are the markers you are given. So when you research these mutation markers that are in your DNA, you can see the regions on a map of where this mutation or recipe change occurred. The DNA is a letter and number sequence given in the place of a name representing an ancestor way back in time. Each mutation is named, and whichever mutations thus spawn from the previous mutation are also named, exactly like a family tree. He begot he, begot he, begot he, and so forth, giving us our unique pathing back in time to the origins of our species. We can see this go back to the cradle of civilization with what is dubbed scientific Adam for the Y DNA and scientific Eve for the X DNA. As our ancestors migrated out of Africa thousands of years ago, and those children branched out and went in different directions, populating the various continents, they passed along in unbroken lines their sex chromosomes. You might find yourself asking, well, if a male can pass along either his Y or X chromosome, why does the female always pass along her mother's X rather than her father's X to her children? And this is a really good question that deserves a thorough answer. So I'm going to link in the description box below 4 informative articles that you should read. The first article is Why Do We Inherit Mitochondrial DNA from Only Our Mothers? By Steph Yin, published to the New York Times in 2016. Second is How Do NonMendelian Genetics Work? By Lindsay Van Sumeren, published to untamedScience.com in 2017. Third, Paternal MTDNA Transmissions on Wikipedia. And fourth, Non-Mendelian Inheritance on Wikipedia. To give the most simplistic overview, upon conception, the paternal mitochondria rapidly breaks down as it arrives in the egg of the mother, and the maternal mitochondria is almost always dominant. In genetics, we have Mendel's laws. They are as follows, and I quote, Mendel's first law, the law of segregation. A parent who has 2 alleles for a gene can only pass on one allele or the other to each offspring. Mendel's second law, the law of independent assortment. 2 or more traits are inherited separately from each other. They don't always occur together. Mendel's third law, the law of dominance. One dominant allele can take charge over a recessive allele and mask it. The only way recessive alleles can be seen is if an individual possesses 2 copies of the recessive allele. End quote. We then have something called non-Mendelian genetics, defined as, quote, any inheritance patterns that don't follow one or more of Mendelian genetics, end quote. So, the fact that our maternal X nearly always overrides the paternal X in female offspring is an example of non-Mendelian genetics. And this is why we always carry our direct maternal haplogroup and not our father's, even though he contributes his X to female offspring. Yet, there can still be an extremely rare occurrence where the paternal X haplogroup is passed along to his offspring and overrides the maternal line X. But I have to emphasize, this is extremely rare. This is called paternal MTDNA transmission. And again, I encourage you to read those articles linked in the description box below. Now, at the start of this video, I'd said that humans have 23 pairs of chromosomes. The key word being pairs. Humans actually have 46 chromosomes. And when we inherit 50% of DNA from our mother and 50% of DNA from our father, essentially we're taking 23 lines of code from each parent for a grand total of 46 chromosomes. As we look at the 23 pairs of chromosomes, we can actually get an idea that these chromosomes have paired off. Chromosome number 1 being half from your mother and half from your father. Chromosome number 2, the same thing. Chromosome number 3, number 4, you see the pattern. These are two parallel sides of a double helix, two strands of genetic data wound around each other. When we read the number sequence of our chromosomes from left to right, we can visualize these strands as being laid out horizontally, so we can read the data. And since we inherited a line of coding from each parent which paired off to create each chromosome, we can visualize two halves, a top and bottom, of each line. One half, usually the top, representing the paternal chromosome data, and the bottom half representing the maternal chromosome data. The data which we inherit randomly comes to us in groupings, long and small, called segments. What does this look like, and how does it help us? So, for example, you have a maternal first cousin. Your common ancestors are your mother's parents, so your maternal grandparents. When we compare our autosomal DNA one-to-one, these matching segments are representing your maternal DNA, ergo, DNA data you inherited from your mother, and they are representing both of your maternal grandparents. So you can now identify these segments of your unique chromosome map as having been inherited from your maternal grandparents. Three things here to note. Number one, I'd said your unique chromosome map. No two chromosome maps will ever be the same. Why? Because of random inheritance. You are the subject of your chromosome map. This is a map of you and the genetic segments you personally have randomly inherited from all of your ancestors in time. If you and the same maternal cousin compare individually to another maternal cousin, not only will this other cousin highlight some of the same places already identified, but additional ones also representing the same set of maternal grandparents, which the other cousin didn't inherit. Additionally, these cousins will have chromosome maps equally unique, again, all due to random inheritance. You have all inherited some of the same and some very different segments of DNA data from the same set of grandparents. And the goal of chromosome mapping is to discover which segments you have inherited from who. Number two, another goal would be to attempt to further divide the segments and identify which of these shared segments came from which grandparent. So we're looking to separate the segments and identify which ones came from your maternal grandfather and which ones came from your maternal grandmother. To do this, we step back an additional generation. Perhaps your grandmother had a sister who would be your great aunt. You would compare DNA one-to-one with either your great aunt or one of her descendants, they being a second or third cousin perhaps, who shares theoretically zero DNA with your grandfather. As a side note, you should consider the possibilities of endogamy between your grandparents and work to compare relations that, to the best of your knowledge, have zero to limited crossover. The more cousins from that line you can compare with, the better. But the segments they match you on should be the same as some of those you've already identified. You can now label them with your grandmother's name or surname, and the segments which do not match are highly probable to have come from your grandfather. This process is repeated, focusing on the common ancestor or apex ancestor shared with a DNA match, as you work your way across your family branches and back in time. And number three, how might this help us? What's the purpose? Imagine you have a DNA match, and for whatever reason, be it they provided no tree, perhaps they were adopted and have no information to give, perhaps there's no response. If you can access their DNA with a chromosome browser and compare them against already identified segments in your chromosome map, you will get a really good idea of where they belong in your tree. This helps you in so many ways. I'm not even sure where to begin to list them all. And one of the best things about chromosome mapping is when you are contacted by someone who was adopted and doesn't know anything about their family history, you may already have a very clear picture of how they connect to you and who your shared ancestors may be. And this is a similar method to how we solve adoption cases with DNA. When dealing with DNA segments, you're going to notice all the numbers. These numbers correspond to measurements. Just as a ruler deals with inches, centimorgans, and millimeters, DNA is measured in morgans, M, and centimorgans, cm. You're mainly going to be concerned with centimorgans. How many total centimorgans you share with someone, that being the length of all the small segments tallied up, as well as the starting and ending location of a specific segment. Here, you're looking at a chart by the Shared Centimorgan Project. Using the centimorgan count you share with a DNA match, you can begin at the self node and scout around for all the potential relationships the CM count may represent. DNA data changes rapidly, and when it comes to making DNA connections, we must stay within the 5 to 7 centimorgan range to have a solid match about 6 to 8 generations back. These are the generally accepted ranges. However, as the saying goes, there are exceptions to every rule. You can use these smaller centimorgan matches when you are more intimate with your tree and have a chromosome map as a guide. So why might you need to be concerned with the starting and ending locations of a specific segment? Join me in a special interview with Wanda Pierce as she explains her chromosome mapping of ancient bloodlines. That video will be linked on screen. Up next, learn about chromosome mapping with Admixture. Thanks for watching, and don't forget to hit the subscribe button and give this video a thumbs up. It helps with the algorithms and is greatly appreciated.